What a good day, Ms. Moira. This is a report entitled Investigations of Exogenous Applications of Carbohydrates on the Growth and Vitality of Live Oaks, authored by Thomas Martinez Trinidada and et al. This is an introduction of our study. Urban trees often grow under stressful conditions, and arborists use various practices to maintain or improve tree health. Carbohydrates, the main source of energy for plants, are recommended as an alternative source of energy. Sugar applications have been recommended as a potential arboricultural practice to improve plant performance or supplement photosynthetic energy. Growth variables such as tree height, trunk diameter, and root growth are used to assess tree vitality.
Soil microorganisms are another important component in the use of carbohydrate application in soil and the impact of vitality. Microbial activity can affect the rate of nutrient uptake by tree root systems, affecting nutrient availability. Applications of carbohydrates to soil as drenches can increase microbial activity in the soil, but also immobilize nutrients in soil and affect growth rates of plants. Evaluating microbial activity after carbohydrates applications and nutrients analysis of soil and leaves will help determine and interpret the effect of continuous carbohydrates application on soil and tree nutritional levels.
Carbohydrates are translocated and stored in different tissues in response to six source. Relationship analysis of carbohydrates content of different tissues has been used to assess the impact of environmental factors or chemical application on trees. Chlorophyll, fluorescence parameters have been used to detect biological stress on stress and the ratio between variable maximal fluorescence FVFM has been used as an indicator of tree vitality.
Method. In 2004, 60 field-grown live oaks were randomly selected from nursery in Monaville, Texas. The trees were originally planted in 1999 and grown under similar conditions. They appeared healthy with no visible signs of natural and cultural stress. Solutions of corn-derived glucose, starch, or 50-50 mixture of each were applied to the soil around the trunk at different concentrations. Trees were separated by at least one untreated tree to serve as a buffer between treatments.
Method. Trunk diameters were recorded each season at 30 cm above the ground using a diameter tape measure. A digital camera was used to collect DIFF formatted images of all trees. Images were analyzed using the digital imaging surface image to quantify canopy density over time. To evaluate root growth, 4 soil cores were extracted in a square pattern at 0.5 meter for the trunk of all trees. Herbicide, glyphosate, was applied periodically to eliminate weeds during the experiment. Soil samples were extracted 1 and 2 years after treatment application using a 15 cm deep by 6 cm diameter core sampler. Root dry rate was measured with a precision scale.
Methods. Twig samples were analyzed for carbohydrate content every 4 months. Glucose and starch content were determined for each sample using sigma GAGO gradients. Net photosynthetic rate was measured using a portable photosynthesis system. Chlorophyll fluorescence was measured every 4 months. Potential carbohydrate uptake by tree under field conditions was determined by carbon in-stock ratio. B13C tissue samples from twig and roots were collected from water control glucose 40 and 20 GL1. The starch treatments at 18 months, capsulus and place P99 well microtrites plates. Soil sample were taken with 0.5 m around the trunk and microbes activities analysis.
Results. This study investigates the impact of high concentration of carbohydrates on healthy, non-stressed trees. Carbohydrate applications as soil drenches can affect tree growth through increasing carbohydrate interchange between the soil and root system, intensifying microbial biomass and sequestering or immobilizing nutrients. The study found no visible phytotoxicity in soil in healthy trees, but rates of carbohydrates use caused no significant visible phytotoxicity. Trunk growth of healthy live oaks was not significantly affected by soil drenches application of glucose, starch, and a 50-50 mixture.
Result, however, sucrose, fructose, and glucose applied at 50 or 70 grams liter 1 resulted in patient growth in Vitola pendola road. The study also found no significant differences among treatment for canopy growth, vitality, or glucose content in twigs. The carbon isotopy ratio D13C was within the range of D13C values for C3 plants, and no evidence that potential sugar uptake can alter the carbon isotopy ratio. So respiration was found to be significantly influenced by carbohydrates and concentration, but no significant increase in tree growth or vitality was detected.
Conclusion: growth can be negatively affected by the carbohydrates application to the soil, as soil microbes can complete and sequester nitrogen in soil. Digital photograph of the monitoring canopy growth showed no significant difference among treatments, suggesting the concentration used in the experience failed to significantly increase canopy growth of healthy live oaks. The study also found no evidence that potential sugar uptake can alter the carbon isotope ratio, as exogenous carbohydrates can from C4 plant with different D13C signatures.